My name is Laura Chambers, and I'll be presenting my research today entitled Antibiotic Therapy Worsens Outcomes Following Primary Treatment in Ovarian Cancer, Potential Role of the Gut Microbiome. I have no disclosures. The gut microbiome is a collection of microorganisms that live within and upon the body's mucosal surfaces. The gut microbiome has many functions. It helps us absorb nutrients, regulates our immune system, and has also been linked to multiple diseases, including obesity, cardiovascular disease, and cancer. In recent years, the gut microbiome has been implicated as a potential modulator for carcinogenesis and also cancer-directed treatment. In order to evaluate the effect of the microbiome upon cancer treatment, researchers have used antibiotics to change the microbiome and then have looked at the differential responses to treatment in preclinical studies. In a study published in Science in 2018, mice with genitourinary malignancy, sarcomas, and melanomas were treated with antibiotics, and they were found to have decreased immunotherapy response and worsened survival compared to controls that did not receive antibiotics. They looked within a correlate of patients who were receiving immunotherapy for lung and genitourinary cancers and found that those that received antibiotics, patients that received antibiotics, had decreased progression-free survival and decreased overall survival compared to controls. In a similar study published in 2019, 196 patients were receiving immunotherapy, and they evaluated for antibiotic treatment prior to immunotherapy. And they found that those that did receive antibiotics had decreased overall survival 2 versus 26 months and a higher likelihood of primary refractory disease. So far, there's been quite a number of studies that have looked at the role of antibiotics and immunotherapy. Studies have also, however, looked at this within traditional chemotherapies, including platinum-based chemo. In a study published in 2013, mice with lymphomas and colon cancers were treated with antibiotics or not, and they were assessed for their survival outcomes. And it was found that mice that received antibiotics had decreased survival compared to controls, and also reduced induction of pro-inflammatory genes and reactive oxygen species. In a similar study, 800 patients that were receiving cyclophosphamide or cisplatin for leukemias and relapsed lymphomas were assessed for whether they received anti-gram-positive antibiotics during chemo. And it was found that receipt of anti-gram-positive antibiotics worsened chemotherapy response and decreased both progression-free survival and overall survival. In women that are diagnosed with advanced ovarian cancer, the first-line treatment is platinum chemotherapy and cytoreductive surgery. But despite these efforts, approximately 80% of women will recur and die of their disease. Efforts to optimize treatment and prevent platinum resistance in ovarian cancer patients is an urgent need. Our hypothesis was altering the gut microbiome through antibiotic treatment would impact ovarian cancer growth and decrease platinum chemotherapy response. And our goal was to look at this within an animal study and a clinical correlation. We did 2 separate animal studies looking at with BLAC6 immunocompetent and NSG immunocompromised mice. Mice were obtained from breeders at 6 weeks of age. They were given antibiotics in their drinking water, which was a cocktail of ampicillin, neomycin, metronidazole, and vancomycin, or control water. Tumors were injected at 8 weeks of age, and mice were followed weekly for transabdominal ultrasounds for tumor growth. At 21 days, mice were assigned to either placebo treatment or IP cisplatin, which was given to them weekly. For our methods, we used a syngeneic mouse model with ID8 and ID8-VEGF cell lines, which are a syngeneic ovarian cancer cell line in 2 different mouse studies with BLAC6 and NSG mice. Tumors were monitored with transabdominal ultrasound, and we assessed the gut microbiome through stool collection and 16S ribosomal DNA extraction. So now I'll present our results. So this is the study. These are the results that demonstrate tumor growth, and the blue is in blue designates antibiotics, and then water is in red. For both the ID8-VEGF and ID8 ID8 cell lines, we see that in the presence of antibiotics, as shown in the blue line, tumor volume was significantly accelerated compared to control animals, as seen in the water curve in red. This graph shows the 4 treatment groups and the subsequent tumor volume. So to orient you to the figure, there's 4 treatment groups: antibiotic cisplatin in green, antibiotic placebo in red, water placebo in blue, and orange. And orange shows water cisplatin, with the tumor volume on the y-axis and the days on the x-axis. The black arrow is when we started cisplatin treatment. So as you can see here, the green and the red lines, which are the antibiotic-treated animals, not only do they have advanced tumor growth, but as you can see, in the presence of antibiotics, there's essentially no response to cisplatin. However, for the water-treated animals in blue and orange, you see that differential effect, with the water cisplatin group having the decreased tumor volume. We saw this exact same phenotype within the ID8 VEGF tumors, with the accelerated tumor growth in the antibiotic-treated groups, and then in the water-treated animals, there was that cisplatin response that we did not see in the presence of antibiotics. We then looked at survival for the mice, again using those same groups, and we found that the antibiotic-treated animals had significantly worse survival, with the antibiotic cisplatin having a median survival of 64 days compared to water cisplatin of 84, and this was statistically significant. And we saw the same effect for the ID8 VEGF cell lines, with the antibiotic-treated animals having significantly worse overall survival. We assessed the gut microbiome using 16S ribosomal sequencing. So here are the 4 treatment groups. And as you can see, this is a measure of the alpha diversity, which is a measure of the number of species and the relative abundance of the gut microbiome. And basically, each one of these different graphs represents a different time point during the study. But in the presence of antibiotics, we markedly changed the gut microbiome, as you can see here. So this graph shows our 16S sequencing. So each one of these large rectangles represents a time point. So baseline of the study, 2 weeks, tumor engraftment, 5 weeks into cisplatin treatment at the end point of the study. Each one of these different colors represents the proportion of bacteria within a treatment group. And then there's 8 treatment groups at the bottom of the study, with water placebo, with the water groups being the ones on the left side and the antibiotic ones being the ones on the right side. So basically this is 8 treatment groups over 5 different time points. And basically this is demonstrating the abundance of bacteria in each different group. So just to point out a couple of things here. The first thing to note is each different color is statistically significant relative to other different colors around. So as the study goes on, to first point out the gamma proteobacteria genus. So here in the beginning of the study, there's essentially, very minimal of these bacteria. But in the presence of antibiotics, we essentially enrich for these populations, with the antibiotic-treated animals having a higher amount of gamma proteobacteria. The other thing to note is with the groups that responded best, so the water cisplatin groups, they have an increased amount of bacteria of the Akkermansia lactobacillus, and then also Bacteroides and Bifidobacterium species. We then aimed to look at this within a patient correlate and used a retrospective cohort study. So we assessed patients treated at Cleveland Clinic with stage 3 and 4 ovarian cancers diagnosed from 09 to 15. All patients underwent surgery and platinum chemotherapy, and we collected information on their patient demographics, oncologic and surgical variables. And then we recorded whether they received antibiotics for at least 48 hours during primary platinum. We also then stratified by gram-positive antibiotics defined as vancomycin, daptomycin, linezolid. We did a Kaplan-Meier analysis for PFS and OS, and then we did multivariate models built based on backward selection. In total, we included 424 patients in the final analysis, with 147 receiving any antibiotics, 48 patients receiving anti-gram-positive antibiotics, with a median follow-up duration of 50.7 months. These are our patient demographics, and just to point out, a couple of different high points here. So there was no difference in age, BMI, ECOG performance status, receipt of neoadjuvant chemotherapy, R0 optimal debulking, stage, serous histology, or time to chemotherapy between the 2 groups. So to present our data, so First, we stratified by antibiotics and no antibiotics, and we found that antibiotic treatment significantly worsens progression-free survival and overall survival. PFS was 23.1 in no antibiotics compared to 17.4 in antibiotic group with a hazard ratio of 1.5, and then overall survival 62.4 versus 45.6 with a hazard ratio of 1.46. When we stratified by gram-positivity, we found that the anti-gram-positive antibiotics did significantly worse, 16.5 versus 17.5 versus 23.1 for PFS, with a hazard ratio of 1.85, and then overall survival was similar, 35 versus 47.7 versus 62.4 months, with a hazard ratio of 1.21. A multivariate analysis, antibiotic treatment increased the risk of death and cancer-specific death, independent of other variables. And then again, on multivariate analysis, controlling for confounders, receipt of anti-gram-positive antibiotics were associated with an increased risk of recurrence and cancer-specific death. So to conclude, antibiotics during platinum chemotherapy in ovarian cancer negatively impact survival in both a preclinical mouse study and a retrospective human study. Our parallel mouse and human data supports that antibiotic-mediated gut dysbiosis may impact response to platinum chemotherapy, and future studies are ongoing to further understand the mechanism behind the gut microbiome and platinum chemotherapy. It has taken a village to do this study, and I'm so thankful for everyone involved, including members of the Rises Lab, specifically Dr. Rises himself, and then also Emily Ezikoff. And then also our amazing clinical faculty within the gynec department at Cleveland Clinic, especially Dr. Vargas and Dr. Mitchner, and then all those that helped with the data collection, and Meng Yao for statistical support.